Welcome. Today, we're diving into cleaning validation in multi-product API facilities. I'm Dr. S.K. Singh, and together we'll explore the challenges and best practices for ensuring cleanliness when multiple products share the same equipment. Let's get started and uncover how to maintain quality and safety in complex manufacturing environments. Cleaning validation is essential, especially in facilities where multiple products share the same equipment. When different active pharmaceutical ingredients, or APIs, are made on the same line, risks multiply. We're talking about product-to-product contamination, leftover cleaning agents, microbial growth, and even regulatory issues. For example, if equipment used for paracetamol isn't cleaned properly before making metformin, residues can carry over. The goal here is clear: prevent contamination and protect patient safety at every step. When it comes to regulatory expectations for cleaning, there are a few key standards we need to keep in mind. ICH Q7 tells us cleaning procedures must be written down, validated, and backed by solid science. Over in the EU, GMP Annex 15 adds that cleaning validation should be risk-based, focusing on the worst-case product, with acceptance criteria clearly justified. The US FDA expects manufacturers to prove cleaning effectiveness with documented evidence. And today, the trend is moving toward health-based exposure limits, rather than the old 10 parts per million rule. Let's walk through the 4 key phases of the cleaning validation lifecycle. First, we design the cleaning process. This means developing clear, effective procedures. Next comes cleaning validation, where we run qualification studies to prove those procedures work. Then, we move to continued verification, monitoring the cleaning's effectiveness during routine use. Finally, periodic review helps us evaluate overall performance and any changes. For example, if equipment or products change, or if the cleaning procedure itself is updated, revalidation is necessary to keep everything on track. When it comes to validation, we focus on the product that poses the greatest challenge, the worst-case product. Here, APIB stands out. It has high potency, poor solubility, and high risk, making it the toughest to validate. We don't validate every product separately. Instead, we use risk-based selection, considering factors like toxicity, solubility, potency, cleanability, and batch size. This approach helps us target the product that truly tests our process, ensuring safety and efficiency without unnecessary work. Before we dive into solutions, we need to be clear on what success looks like. Establishing acceptance criteria means setting the specific conditions that must be met for a project or task to be considered complete. These criteria act as a shared agreement, a checklist that guides our work and helps avoid misunderstandings. When everyone knows the rules up front, we save time, reduce friction, and keep our focus sharp. It's a simple step, but it makes all the difference. Let's talk about how we calculate the surface residue limit. It's all about figuring out how much residue is acceptable per square centimeter of equipment surface. We start with the MACO, that's the maximum allowable carryover, and divide it by the total product contact surface area. For example, if the MACO is 1 million milligrams and the surface area is 50,000 square centimeters, the limit works out to 20 milligrams per square centimeter. The residue must stay below this to pass. This method gives us a cleaning limit that's solidly based on science. Let's look at an example of swab sampling in cleaning validation. Here, a 100 square centimeter area was swabbed, and 0.5 milligrams of residue was recovered. That gives us a result of 0.005 milligrams per square centimeter. Since our acceptance limit is 0.02, this passes comfortably. Swab sampling works well for hard-to-clean spots and lets us evaluate the surface directly. For larger vessels or complex piping, rinse sampling is usually the better choice. Each method fits different needs. Let's talk about recovery studies and analytical validation, a crucial part of cleaning validation. We need to show how efficiently we can recover residues from surfaces. That's where the recovery factor comes in. It's calculated by dividing the recovered amount by the applied amount, then multiplying by 100 to get a percentage. For example, if we apply 100 micrograms of residue and recover 85 micrograms, that's an 85% recovery. Generally, anything above 70% is acceptable, as long as we have solid scientific reasoning. Common analytical methods include HPLC, TOC analysis, UV spectroscopy, and other specific residue tests. We've successfully completed the validation process. This slide shows the results from 3 consecutive runs. Each one passed without issue. The protocol we followed is thorough, covering everything from the objective and equipment details to cleaning steps and where samples were taken. It also defines clear acceptance criteria and the analytical methods used. Typically, we require at least 3 successful cleaning cycles in a row to confirm the process is reliable. This ensures consistency and confidence in our cleaning validation. When regulators review our processes, they often find common gaps that can hold us back. These include not fully justifying the worst-case scenarios, skipping critical calculations like HBEL or PDE, and missing key sampling spots. Sometimes recovery studies aren't thorough enough, or dirty hold times aren't validated properly. Change control can also fall short. For example, using a highly soluble product for validation instead of the toughest-to-clean API raises red flags, because it doesn't truly reflect the worst-case conditions we need to prepare for. When it comes to best practices, we focus on setting acceptance limits based on health-based exposure levels, and we use risk-based strategies to guide validation. We always plan for the worst-case product scenario, back up our sampling with solid science, and keep documentation thorough and clear. Quality risk management is integrated throughout. For cleaning verification, routine monitoring is key, from visual checks and swab tests to trend analysis and regular reviews. Emerging technologies like automated cleaning systems, online monitoring, and AI-driven optimization are shaping the future of our processes. Cleaning validation in multi-product API facilities is all about proving, with solid documentation, that cleaning procedures work every time. They must remove residues, contaminants, cleaning agents, and microorganisms, down to levels that science supports. This process isn't just best practice. It's required by a range of regulations and guidelines, from the FDA's CGMP rules to EU GMP, WHO standards, and several key international frameworks. Together, these ensure safety and quality across the board. Thanks for joining me today. If you want to explore this topic further or have any questions, please don't hesitate to reach out. I appreciate your time and attention, and I look forward to continuing the conversation. Thank you. This video was created with SlideSpeak. Visit SlideSpeak to turn your presentations into engaging AI-narrated videos.